Dear students, as we have discussed earlier about the gene bank and the cryo bank, another bank is seed bank, means stored the seeds to preserve the genetic diversity. So you can say it is a type of gene bank. There are many reasons to store the seeds. First is to preserve the genes that plant breeders need to increase the yield for breeding, or you can say for hybridization or cross, for disease resistant, drought resistant, nutritional quality, taste, etc. of the crop. The second one is to forestall loss of genetic diversity. A rare or imperiled plant species in an effort to conserve the biodiversity ex situ. Many plants that were used centuries ago by humans are used less frequently now, so the seed banks offer a way to preserve that historical and cultural value. Collection of seeds stored at constant low temperature and low moisture are guarded against loss of genetic resources that are otherwise mentioned in situ or in field collections. These alternative living collections can be damaged by natural disasters, outbreaks of disease, or war. Seed banks are considered seed libraries. You can say the library is the in library we kept the books, and the seed bank we kept the seeds containing valuable information about evolved strategies to combat plant stress that can be used to create genetically modified versions of existing seeds. The work of seed banks span decades and even centuries. Most seed banks are publicly funded, and seeds are usually available for research that benefit the public. So now you have to read out the storage conditions and regeneration. Generally, seeds are kept at adjusting or required minimum moisture and temperature. The mother plant, many seeds attain an innate ability to survive drying. Survival of these so-called the orthodox seeds can be extended by dry, low temperature storage. The level of dryness and coldness depend mostly on the longevity that is required and the investment in infrastructure that is affordable. Practical guidelines from a US scientist in 1950s and 1960s, James Harrington, are known as thumb rules. The hundreds scientists in the 1950s and 1960s rules, the 100 rules guides that the sum of relative humidity and temperature in Fahrenheit should be less than 100 for the sample of survive for 5 years. Another rule is that, so you can write down the rules for storage condition. Another rule is that the reduction of water content by 1% of temperature to degrees Fahrenheit will double the seed life span. Research from the 1990s showed that there is a limit to the beneficial effect of drying or cooling, so it must not be overdone. Understanding the effect of water content and temperature on seed longevity, the Food and Agriculture Division of the United Nations and consultancy group called Bioversity International developed as a set standard of international seed banks to preserve seed longevity. The document advocates drying seeds to about 20% relative humidity, sealing seeds in high quality moisture proof convention storage protocols, seeds from our most important species corn, wheat, rice, soybean, pea, tomato, broccoli, melon, sunflower, etc. can be stored in this way. However, there are many species that produces seeds that do not survive the drying or low temperature of conventional storage. Coconut, papaya, oak, walnut, and willow are a few examples of species that should be preserved cryogenically at a very low temperature. So you are all directed to read out and point out the important lines, and in your examination there may be a question on this topic. They may ask a short note on the seed banks. Okay, I hope you are learning the topics earlier sent for you in the form of PDF. Now I am reading or trying to make it clear for you, so you all maintain your continuity of the reading and learning process of your study and the contents given in your syllabus. Okay, thank you.